watch this video that we've never seen before. Okay? I'm going to stop it quite frequently, all right, just to hammer home some points with you folks.
This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system.
Where do we normally see the neuromuscular junction? That's what this is showing us. Would we find that at the end of the muscle or in the middle of the muscle? Yeah, mid-region. Okay, you're gonna find these neuromuscular junctions normally in the middle portion of the muscle, or right around that area. Yep.
A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres.
Just a quick review here, okay? So in the middle of our sarcomere is, the ends of the sarcomere are delineated by the Z discs, okay? Z lines, right? So it goes from Z line to Z line, and that's a sarcomere. Right? And the center is what? What do we call this center line here? M line, yeah, think of it, middle line, okay? Right? So what is anchored to the M line? Easy to remember because it starts with the same letter. Myosin, the thick filaments. The thick filaments are anchored to the myosin, okay? And then on the Z discs, what's anchored to the Z disc? Actin, thin filaments, okay? Thin filaments are anchored to the Z disc. Now on either side of the Z disc, we have this little region here, okay? So where it's a little bit lighter, okay, only because there's thin filaments there, that region is called the I band, okay? So it's this area here that gives the light appearance to striated muscle. You remember how striated muscle has alternating light and dark regions, right? So this is what helps to give it that light appearance because there's only actin present in that area there, okay? This region will narrow significantly and quite possibly disappear during a muscle contraction, okay? So on either side of the M line, okay, you got this phenomenon just where the myosin is present. There's no overlapping in this area. And that's called the H zone. The H zone only has myosin present, and it will also narrow or possibly disappear during a muscle contraction. So when we see this thing start to contract, you'll see that these zones here will start to significantly decrease in size. They don't totally disappear, but that's what we're going to see there, okay? All right.
Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments.
True or false? The length of actin and myosin becomes shorter, okay, with the muscle contraction. Is that true? That's right. They never change. The length of actin and myosin never changes. The amount of overlap does, okay? They never change. Okay? That's a great true-false question.
Although the action between the filaments is described as sliding.
Sorry, I have to see what somebody said. Give me 1 second. Yes, they do look, yes, that is correct, right? And especially during overlap. What did I do? Oh, no, wait, here we go. What did I do? All right, here we go.
The myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle.
But also that's known as the power stroke. Remember the 4 steps to the actual cross bridge mechanism? Okay, the first is when we form the cross bridge, that's step 1. Okay, and then the second is going to be the power stroke when the myosin head swivels and pulls, right, the actin past it. Okay, and then it has to release. Okay, and we'll talk about that in a second. Right, and what's needed? Do you remember what we need to release the myosin head from the actin? ATP. Okay, the ATP allows it to release, and then, okay, we have to cock it again. Okay, so that's the cocking part, and when that cocks, that's when the ATP gets broken down into ADP and the inorganic phosphate. We'll see it here in a second.
Muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. Contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke.
That's important. Okay, so as the power stroke occurs, that ADP and that inorganic phosphate pops off. So now we have that exposed ATP binding site on the myosin head. Okay? Otherwise, we can't bind ATP onto that because the ADP and the inorganic phosphate are there. Right? What's the name of the enzyme that sits on the myosin head and it breaks down ATP into ADP? ATPase. Okay, so it's just remember what I said. ASE, that's on the end of a word, that's an enzyme. Okay, so all we did was just we named the enzyme after the molecule. So ATPase is that molecule, that enzyme that's going to break the ATP down into ADP. Okay.
The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present,
2 things that you need for muscle contraction: calcium and ATP. You need to know that. Right? If you're deficient in either one of those, there's a good possibility that you're not going to be able to generate a muscle contraction. Okay?
calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract.
Okay, just a quick review of what we're going to see here. I hope that everybody has a pretty decent understanding about this. All right, our action potential is going to travel down, come to the synaptic knob. It's going to open up what? Triggers the opening of a certain gate. What do we call those gates? Voltage-gated calcium channels. Those voltage-gated calcium channels will open up. Since calcium is in higher concentration outside of the synaptic knob than inside the cell, it's going to diffuse into the synaptic knob. What creates that concentration gradient? What makes calcium higher outside the knob than inside the knob? What does it in the neuron? Remember the sodium potassium ion pumps in the neuron? They pump sodium out of the cell and then pump potassium in, right? Well, in the synaptic knob, you've got calcium ion pumps, and they're constantly pumping calcium out of the knob, making it higher out of the knob than inside the knob. So once the calcium, voltage-gated calcium channels open up, calcium comes in, it binds onto the synaptic vesicles. Synaptic vesicles release acetylcholine, all right, through exocytosis into the synaptic cleft here. And then you're going to see it here. It's going to migrate across to the receptors here. Acetylcholine is going to trigger the receptors to open up, and it's going to open up sodium voltage gates and potassium voltage gates, right? And sodium will flow into the cell, potassium leaves the cell, right? And that's going to create what we call an EPP, an end-plate potential, right? It's like a graded potential, very similar. It's local, all right? Doesn't last long, it's short-lived. But if it's enough, right, we can reach threshold, and it's going to depolarize the sarcolemma, plasma membrane of a muscle. When that happens, we get our action potential. Same scenario. It's all or nothing. Once that occurs, game on. Okay? The action potential is going to spread out across the sarcolemma, and it's going to go down into T tubules. It's going to look for the voltage-sensitive calcium channels. It's going to open those up. And when those open up, they trigger the opening of calcium channels in our sarcoplasmic reticulum terminal cisterna. Remember that sleeve-like structure that sits on the myofibril there? And that's where the calcium is stored. And then it releases calcium into the sarcoplasmic reticulum, into the sarcoplasm, which we're going to see right here.
Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow.
Saw that? See how those molecules were in the purple tubes here, and now they're here on these sarcomeres? That's representing the sarcoplasm, and that's where your actin and myosin lives. Okay, that is separate. Okay, and you can see now where it's going
to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
All right, that'll be the last time that you have to watch that video with me, okay? I hope it's helpful. All right, let's bump out of that. And all right, let's finish up chapter 10. We're going to talk about the aging effect on muscles, and then we're going to quickly go through cardiac muscle and smooth muscle. All right, it's not a lot because you're going to hit both of those muscle tissues in more detail in 211. Okay, so we're going to finish up aging. And then we'll talk about cardiac muscle and smooth muscle. Where are we? There we go. Effects of aging. KB. All right. So, a couple things that you may have heard me talk about before in chapter, I think it was chapter 5 with tissues. One of the things that you'll notice is, well, you won't notice it because most of you are pretty young in regards to this. But by the time you get into your 30s, possibly even your 40s, you are going to start to decrease in general your own activity. Your skeletal muscle, all right, tissue is going to start to decrease. And they say roughly by your 3rd, 4th decade of life, you start to lose every decade after that about 10% of your muscle mass. Now that varies. Right? If you're an active person, obviously you can slow that down, if not prevent it. Okay? And there's certain extreme cases. So when you start to decrease your muscle mass, you're going to see a decrease. And obviously, the power, the tension, the muscular tension, the force of a muscle contraction, right? And if you're not as active as you once were, remember the saying, if you don't use it, you lose it, okay? We start to experience muscle atrophy. So the size of the muscle will decrease. If you're decreasing the power and the size, you're going to decrease the muscular, sound like Popeye, muscular endurance, okay? So as your muscle size decreases, you'll start to decrease the amount of muscle fibers that you have, and your muscles will actually start to, overall, the diameter will decrease, right? And when all these effects occur, okay, you'll lose the loss of myoglobin, so your oxygen storage capability decreases, mitochondria count decreases, right? And as you get older, your circulatory system doesn't circulate the blood as well. So you start to decrease the amount of blood flow, right, to the muscles, right? Now again, that's if you're pretty much a sedentary person, if you're not doing much. If you're an active person and still promoting activity, you can prevent a lot of these effects from occurring here, all right? So when you do injure yourself, and I know I didn't talk about the satellite cells too much, but they're like the muscle cells version of glial cells, right? They're support cells, right? So if a muscle cell gets damaged, the satellite cells help to repair them, okay? So as we get older, we start to see a decreased number of satellite cells, so that therefore it impairs the ability of the muscles to help repair themselves. And a lot of times when a muscle gets damaged, all right, and you don't have a satellite cell there to help repair it, then fibrosis occurs. And remember fibrosis when we were talking about tissues in the skin there, that's scar tissue, right? And so instead of us repairing a muscle cell with muscle tissue, okay, we're actually filling it with connective tissue. Connective tissue doesn't have the same characteristics and properties as muscles. Okay? It's not conductive, it's not elastic, it can't contract, right? So we start to lose because we're replacing that muscle tissue with that fibrotic tissue, and that's dense connective tissue. Okay, so it can't stretch, and therefore you don't have that flexibility like you did before. So now you're actually promoting the possibility of making it easier to injure yourself. Okay, all right. Where am I? This right here is just pretty much, I don't know why I have like 2 slides that are the same. This is pretty much what we just talked about. Okay, so I'm just going to pop by that one. All right, so what are some of the changes in skeletal muscle and accompanying aging? Okay, like I said, okay, decrease in muscle mass, okay, due to decrease in fiber number, muscle fibers decrease. Because the fiber number decreases, so does the size of the muscle, the diameter, right? Do lose some of that oxygen storage capability, okay? So the myoglobin is now not present as it once was. Decrease our vascular supply to the muscles, right? Satellite cell numbers decrease, so when you injure yourself, it's replaced with fibrotic material instead of muscle tissue, right? And so you lose that elasticity of the muscles, right? And this happens over time. Okay, so let's jump into cardiac muscle, okay? Now remember back when we were learning about some of this tissue in lab, all right? We learned that skeletal muscle, those muscle cells are long and cylindrical, and they'll have many nuclei, okay? So with cardiac muscles, right, they're not going to be as long as skeletal muscles. They will be striated, okay, because they also have sarcomeres. Okay, remember the sarcomeres give the muscle tissue its striated appearance. Okay, cardiac muscle cells can have more than 1 nuclei. They can have 2. Okay, but I always think of cardiac muscle cells as like a Y on its side. Okay, so there's a branching, but they're not very long. Right, but think about this: your heart muscle is beating all the time. Has anyone ever heard the theory that you only have so many beats in your heart? I heard a story the other day, and I've heard it on many occasions. There's even jokes made about it, about certain people won't exercise because they don't want to use up their beats. But there was a guy, he was a mechanic, and he said there's only so many, in your fuel pump, there's only so many times that it's going to pump fuel, and then it's going to honk out. Well, this same person believes the same thing about their heart. So if you exercise, you increase your heart rate, your heart beats, and so you're going to burn through the number of beats that your heart is actually allowed. Anyways, if you think about it, muscle, all right, cardiac muscle needs an endless supply of ATP, pretty much, if you think about it, because it's always beating, okay? When you're sleeping, when you're awake, when you're exercising, when you're resting, it's always beating. So therefore, it only makes sense that we have tons of mitochondria in this tissue, okay? Because, right, when mitochondria are present, then you're going to undergo aerobic respiration. Okay, so when you break down one molecule of glucose, you'll get 32 ATP. Because if you're not undergoing aerobic respiration, then one molecule of glucose is going to net you 2 ATP. That sucks. Okay, so you'd be burning through a lot of glucose. Okay, so we're going to have that mitochondria. We're also going to have these guys. You guys remember the intercalated discs? Okay, those are those desmosomes and gap junctions. Basically, what those do is, one, desmosomes help to anchor the cardiac muscle cells to one another, so they can have a much more effective contraction and relaxation. But also, the gap junctions create these holes in between muscle cells. If you know anything about electricity, okay, when you have that electrical discharge along that action potential, right, that electrical current wants to go. Electricity is always going to want to go to the area of least resistance. Okay? Same principle applies here. These gap junctions allow that electrical activity to wash over the cardiac tissue quicker and easier, because the electrical activity can move through those gap junctions and then stimulate the neighboring cardiac muscle cell. Okay? So one of the things that you need to understand, and I'm sure you've heard about this before, the pacemaker, right? Your cardiac muscle cells have what we call an autorhythmic pacemaker. Okay, so they will stimulate the muscle cells to contract. All right, so if you've ever seen the movie Indiana Jones and the Temple of Doom, has anyone here ever seen that movie? Right, there's a scene in which the guy reaches into people's chests and he pulls out their still beating heart. There's some truth to that, right? Mainly, the heart will beat on its own for so long, okay, because it has this innate rhythm to it. It's your nervous system that regulates your heart rate. You can speed it up or slow it down. Right? If you were to take all the nerves out, that heart will still beat until it flows through all of its, fuel, and it will stop. Right? So what allows the heart rate to be influenced, and we'll talk about this a little bit more in later chapters, all right, is the autonomic nervous system, the ANS. That's your autopilot nervous system. Okay? Like I said, heart rate will be influenced, either increased or decreased, by the nervous system, specifically the autonomic nervous system. Okay, so the rate of contraction, and not only that, but the force, because it can actually increase how hard that muscle can contract. Okay, but you get to learn all that fun stuff in 211. Okay, so here's a nice picture of our cardiac muscle. Okay, so you can see we got our branching going on. Right, it's striated because we have sarcomeres here. Tons of mitochondria all throughout. I'll even zoom in here a little bit. Okay, so you can see here are the gap junctions. So the electrical current can flow through these structures, right, thus spreading out the electrical signal much easier. Then you have these desmosomes here that anchor one cell to the neighboring cell. Okay, and that allows for more efficient contraction. Okay. Here's one of our nuclei. So very similar, right? And then you can see the intercalated discs are basically all those desmosomes and gap junctions. Okay. All right, so this slide is a really good slide because it shows you some of the differences between cardiac muscle and skeletal muscle, right? Yes, they both have more than one nuclei, except cardiac muscle can have up to 2, where skeletal muscle can have many more, right? Skeletal does not branch, cardiac does branch. Okay? The skeletal muscle, right, is going to be much longer. You can have some of these cells be as long as the entire length of the muscle. That's quite impressive. The cardiac muscle is much shorter. Okay? But it will be a hardier cell or a thicker cell. Okay? So no intercalated disc in skeletal muscle. Keep this in mind. Cardiac muscle is stimulated by the autorhythmic pacemaker. So it could just beat on its own, okay? Whereas skeletal muscle is going to be stimulated by the nervous system, but not just the nervous system, the somatic nervous system. It's the voluntary nervous system, okay? All right. See? That's it for cardiac. You'll go into a little bit more detail for cardiac. All right, let's wrap up with smooth muscle, okay? So smooth muscle, right, there's a reason why we call it smooth, right? Because there is no sarcomere in smooth muscle. Okay, so you will not have that striated appearance. Okay, so we're going to see smooth muscle in several organ systems, and here's just a couple. In our blood vessels, okay, because remember, smooth muscle is going to control the diameter of a blood vessel. You can make it bigger or smaller. If it's smaller, it's harder for blood to flow in it. It'll decrease the blood flow. If it's bigger, it allows more blood to flow through it, okay? So therefore, it can regulate blood pressure, okay? Not only that, but also the flow of blood. Same kind of scenario goes on in our bronchioles, in your respiratory system, okay? So the air tubes. If you increase the diameter of the air tube, more air can flow in. If you decrease it, like in an asthmatic, they're having an asthmatic attack, the bronchioles constrict, or bronchoconstriction, all right, then it increases the resistance to the airflow, okay? So smooth muscle can regulate airflow, right? Ideally, you want to get it down to the alveoli. Alveoli are those little balloons at the base of the bronchioles. So you'll have, here's your bronchiole, then you'll have the alveoli, your air sac there, okay? So if it shrinks down, if it gets really small, okay, it's going to greatly limit the amount of air that can get down in here, okay? You want it big, like this, okay? Intestines, same thing. That's how we move food product through your bowels, okay? Smooth muscle will contract and relax. It's called peristalsis, right? It'll move products through your intestine, okay? Your stomach too. You have 3 different layers of muscle, and they're arranged in different orientations so they can help mix up the food inside your stomach and help move the food out, okay? So also that's what's going to happen in your intestines at the same time too. It's going to move it. As it's moving it, it'll mix it, okay? So it gets exposed. All right, urinary system, same type of situation. All right, how we are going to get urine, all right, from the kidneys down to the bladder. All right, then we'll also see it in the female reproductive system. You'll learn about that when you talk about the uterus and whatnot. Okay, there's different layers in the uterus there. All right, that's just a picture of that. So let's quickly just kind of talk about a smooth muscle. Okay, I describe it as a football-shaped cell. The book calls it a fusiform, like a fibroblast. Okay, it's going to look like this. It's actually not a bad drawing. I'm very proud of myself. All right, so all right. In the middle, it's going to be wider. At the ends, it'll be tapered like a football. Okay, so there is one nucleus. Okay, it doesn't have more than one. It's just one. Okay, so our sarcolemma, that's the plasma membrane of a smooth muscle, has this calcium channels here. Okay, so there are chemical calcium channels, voltage calcium channels. All right, we do not have T-tubules. T-tubules do not exist. Okay, instead we have alveoli. I don't know why my voice is making this raspy noise. I think I was smoking a cigar before. Okay, so a calveoli is this invagination, but it looks more like a flask, kind of like this. Right? Whereas the T-tubule just kind of looks like this, okay, like a well. Okay. Similar type of scenario here. We still have a sarcoplasmic reticulum, right? So you'll have some calcium there, but much more important than that is the calcium that's outside the cell, hence the calveoli. Okay, so quick picture here. If you look here, all right, this is one whole smooth muscle cell. All right, so on the sarcoplasm, on the sarcolemma here, you've got these little invaginations here. Okay, and that's the calveoli. Right, and so we won't really see our sarcoplasmic reticulum like we saw before. Okay, on the inside here, right, you're going to see these structures here. Where's my picture? Hold on. This is the one I'm looking for. There we go. All right, let me pop that back out. Okay, so we've got our calcium outside, right? And then we have our invaginations here, the calveoli here. So in the calveoli, we have our voltage-gated calcium channels, right? So obviously, right, when the muscle cell is stimulated, okay, action potential triggers the opening of the voltage-gated calcium channel. Calcium enters in. Now you've seen this protein before, calmodulin, right? Calmodulin and calsequestrin, we saw those are storage proteins for calcium found in the terminal cisterna in the sarcoplasmic reticulum. I briefly mentioned it. You may not have remembered it, okay? So what happens is that calcium binds onto it and causes a configuration shape change here, right? And then when that occurs, we call that the calcium calmodulin complex. So what it does is that it stimulates, right, what we call the myosin light chain kinase. Remember, kinase is an enzyme, okay, that likes to stick phosphate on it, okay? So it's inactivated. So what happens is this calcium calmodulin complex comes over, finds this enzyme here, activates it, okay? And then this enzyme, remember I told you, it's got kinase on it, so it phosphorylates, it sticks phosphate onto stuff, okay? So what it's going to do is it's going to take that phosphate and it's going to jam it on to the myosin head here. And when it does this, it activates the myosin head, and you guys know the rest of the story, right? We're going to start to get that myosin head connecting to the actin and getting our contraction, okay? So it's a little bit of a different process. So that's why smooth muscle kind of throws you for a loop. You already know what occurs in skeletal muscle and in cardiac muscle, okay, because they have sarcomeres. So that story doesn't change. A little bit different here. So just a kind of quick review, okay, because we're not dealing with, you know, the sarcoplasmic reticulum like we were before, okay? So primarily, what we're going to see is that calcium's outside, right? Voltage-gated calcium channel opens up, calcium rushes in, right? It interacts with the calmodulin, okay, which is a calcium protein. It causes a configuration. You can see how it changes the shape of it, okay, when it binds onto the calmodulin. And that calmodulin is now going to activate this enzyme. It's inactivated, okay? Normally, when the muscle's at rest, it's inactivated. It's not doing it, okay? So we need that calmodulin with its calcium to activate this enzyme. And then that enzyme comes over, and it's going to break down the ATP and take that phosphate, stick it onto the myosin head, and that's going to cause, right, our contraction, our power stroke and whatnot. Okay, so it's a little bit different, okay? All right, I know I jumped ahead there. So you're going to kind of see, right, we don't have our sarcomeres, but we have these structures here, these what we call these dense plaques here, okay? So when we're in our relaxed muscle here, okay, you still have your actin and your myosin. So here's your myosin, your thick filament, and then you got your actin molecules here on either side, okay? So like I said, when that myosin, when that myosin light chain kinase comes over and it phosphorylates the myosin head here, we're going to see, if you look, see how big this triangle is here, okay? We're going to get a power stroke. And this is what happens. Look at the size of that triangle now. It's much smaller. Big, small. So it's like here's the cell at rest, okay? And when it undergoes, all right, a contraction, it looks like this. See how the cells get smaller and they kind of twist a little bit, okay? That's how we're going to get our contraction. Have you ever seen, this always reminds me if you've ever bought beef at a butcher and they put that net around the beef. Have you ever, some people don't know, but if you ever gone to a butcher and there's certain meats and the beef, there's a tight net on it and it looks like that. That's what it looks like to me. All right. Okay, yeah, that's an even better one. More people, especially if you're a vegetarian, not saying that you are, but yeah, most vegetarians are like, what are you talking about? So let's talk about, just quickly, I know I showed you in pictures, but let me just throw this out at you, okay? So when we're dealing with, all right, smooth muscle, okay, we're going to have these anchoring proteins, right? So these anchoring proteins are going to be attached, or excuse me, I should rephrase that. Our actin and myosin will attach to these anchoring proteins here, okay? And so the points where these proteins interact with one another are what we call dense bodies, all right? And then we also have our dense plaques. Point being is, and this is, I don't want to get into this in too much detail here because it's kind of boring and whatnot. But when we're dealing with our contractile proteins, when we start to see a contraction occurring, all right, we change, all right, this distance between those dense bodies and those plaques. That's the okay. All right, that's these guys right here. See the dense bodies here, how far they are from one another. Okay, so when we get that power stroke occurring, now the distance is shortened between those dense bodies, right? And that's what causes, all right, the cell to get smaller, and it's contraction there. Okay. So this type of contraction is what we refer to as the latch bridge mechanism. Okay. This latch bridge mechanism, which is a nice feature to have because it allows us to have contractions for an extended period of time without having to utilize more ATP. Okay? So the players, just a quick reminder, calmodulin, that's the protein that's going to bind calcium, and that's going to change the shape of that calmodulin. And that calmodulin is then going to activate our myosin light chain kinase, right? And that's what's going to phosphorylate or add that phosphate to the myosin head, right? And then that will allow for the contraction, okay? The enzyme that stops the contraction is going to be the myosin light chain phosphatase. Again, phosphatase, this enzyme is going to pop that phosphate off the myosin head. When it does that, it creates relaxation. Okay, so it's a little bit different here. So one of the important things that you need to understand about smooth muscle is this guy right here, and it's fatigue resistant. Okay, so in comparison to skeletal muscle, it doesn't use as much ATP because of that latch bridge mechanism there, right? So it can actually hold its contraction without ATP. It can do so for an extended period of time, right? If you think about it, right, your stomach is constantly going to be churning, especially after a meal. It's going to be mixing and contracting and relaxing and turning that food, and eventually it's going to pump it out, okay? That's going to be important, especially in the digestive tract. I can't wait for you guys to learn about the digestive system. It's weird. It's one of my favorite systems to teach. And I figured I'd like the heart, but actually, the immune system is one of my favorites to teach because that one's fun. I just read an article today that apparently there's a paper out in England that they're saying that there's a possibility that there's no long-term immunity to COVID. Anyone heard that? This came out of England right now, which blows my mind because they said that the antibodies won't last very long. But every time I read these articles, they don't give me the specifics. They say it has something to do with T cells and B cells. That's it. They don't explain to us why it is that the antibodies for this particular virus don't last as long as it would for the rhinovirus or the adenovirus or any other thing. So I'm still looking. If I find anything, I'll let you know, okay? Just to let you know. Okay, so keep that in mind. This is a huge, important characteristic of smooth muscle that when compared to skeletal, it's technically fatigue resistant. It can hold a contraction, virtually uses very little ATP in comparison to skeletal muscle. Okay, so when you get into the digestive system, some of the other chapters in 211, you'll kind of revisit this. Okay, all right, that's it. Let's call it quits for the muscles. I'm ready to get into the brain. Who else is ready to get into the brain? No? Trust me, the brain stuff. What? You know, my sister, she became a social worker, but she had to take a lot of psychotic classes. She had to take a lot of psychiatry classes. And I always tell her, don't try to psychoanalyze me. I can tell when she's trying to psychoanalyze me. I won't let her. So let's jump into the brain here. I'm going to stop recording.